Good afternoon, talks. This is Haiyan Huang, who's from UC Berkeley. She's today going to talk us about gene interactions beyond standard models. So please.
Thank you. Thanks for coming to the first session this afternoon. My today's talk is a brief introduction of some of my recent works related to gene association network. The inference of gene association network is a quite popular topic in system biology for several years, for many years already, actually. And roughly, we can consider the problem involves 3 steps. So the first step is about data preprocessing. So this is actually maybe the most critical step if you want to ensure your whole analysis being appropriate. But I'm not focusing on that in today's talk. At the second stage, in biological network, we don't really observe a network. So we are only able to observe the activities of individual genes. And we hope to, based on the measurements of their individual activities, and some of the individual activities may be reflecting cooperative behavior in some way, then we are able to infer gene network. And at the third stage, after the inference of the interaction edges or the network, then you can work on the community detection. So we have heard a lot of beautiful talks on theories and methods on the community detection already. So my focus today is kind of complementary to that. So I'm talking about the studies we have worked on for how to infer the network or how to assign the edges in a gene association network by using the gene expression data, or the measurement of gene activities across time or over several different conditions. So that's the focus of my today. And for this problem, again, there is a huge and very rich literature on that. So the simplest and also the most commonly used correlation measure people have been using, of course, is Pearson correlation. So like for any 2 genes, we have their expression profiles. We can simply calculate the Pearson correlation to assess their association. So the Pearson correlation is a powerful tool in many situations, and actually in most cases, if genes are truly interacting directly, so their Pearson correlations will be high. So what's the problem of Pearson correlation in practice usually? So we have so many genes, but the time points we have on their expression measurements or the conditions we have on their experimental expression measurements will not be many. So this means that in also very, very often situations, many cases, 2 genes, they are not interacting at all. They are still showing very high Pearson correlations. So we have a very serious false positive problem. And that is the drawback, for using a Pearson correlation. Another situation is that we know Pearson correlation is about marginal relationship. And if we have genes are interacting in groups, so like we have several genes, they are working together in some way. So the marginal correlation for some genes in that group may not be high. And if we are using Pearson correlation, so we may miss that relationship too. Because of that, so people realize that it's important to consider the relationship between 2 genes by taking into account the influence of other genes. They may be all involving in the same pathway or in the same network. And that is the inverse correlation matrix and the partial correlation have been used for. But for the partial correlation as inference, it's appealing in a way that we are able to assess conditional dependency between genes by taking into account the influence of other genes. But again, we need to have the appropriate set of genes to condition on to make sure that inference is meaningful. And usually in practice, we may not have the prior knowledge on the pathway or on the network. Then what we can do in that kind of situation. And another situation in practice we may want to pay attention to is that we may have a huge collection of experiments, maybe involving multiple conditions. Even just for time course data, sometimes the network or the gene behavior may change a long time. In other words, when we talk about networks, we may want to consider the dynamic features. So the gene interactions, whether the interactions are existing or not, or what's the direction of the interaction. So all of those kind of features can just change along time or along the change of the conditions. So we may also want to think about that when we are doing the inference. So I will, as I said, that I want to give a brief introduction on several works we have done along those kind of issues. So the first work I want to talk about is related to the partial correlation inference. But we do want to consider the case that we want to get the partial correlation estimated under conditioning on the appropriate side of genes, even though that prior information may not be known, before the inference. And the second work I want to talk about is related to the local interaction property, as I just have mentioned. So if we have a large collection of experiments, but if the relationship we are trying to study or we are trying to infer, are only appearing in a subset of that experiment, then what we can do. And the last one is actually an ongoing work. So this one here, we want to study the change of the network relevant to phenotype or genotype variation. So since this is ongoing work, I wouldn't be able to give any details about the methodology. I will only want to talk about, conceptualize, what we want to do. So you can see that these three works are, I'm jointly working with Rachel Wang and Peter Bickel on a lot of this work, and both of them are in the audience. And the biologist collaborators are Kenny and Lewis for the first one, and also Medina and Beth at Children's Hospital for the last one. And the second one was a joint discussion. Oh, the idea was coming from a joint discussion with Michael Wortman, several years ago. Okay, so for the first one I want to talk about is related to the partial correlation inference. And when we talk about partial correlation inference, of course, we need to mention the Gaussian graphical model. So everyone, here should be very familiar with this already. So basically, I just want to point again that the partial correlation is proportional to the element, in the inverse of occurrence matrix. And there have been huge efforts, played on already on the inference of this inverse of occurrence matrix, especially thinking about the sparsity of the matrix, and also how to really get the appropriate conditional set, to work on. So I particularly want to talk about this paper, for the scaling up purpose. So this is the work by Menshausen and Billman in 2006. So in this one here, so they noticed the problem, as I have mentioned, that for the inference of the partial correlation, we do want to get the right site to consider. And what they are doing is that, so the main formula here is based on this objective function. So you see that, so the XA here can be considered as the expression profile for gene A. And then they are looking for a linear combination of expression profiles of other genes that can be approximating, the expression profile of gene A, in a good way. So the good way, of course, is measurement by this kind of norm, and also we do want to penalize, the coefficients in the linear combination to make sure the sparse, results. So this one here is based on the assumption, of course, for every gene, maybe only a small set of other genes will be relevant, and this helps to identify that. So our work is closely relevant, idea-wise, to this one. But what we are considering is, in this study here, so they are doing this study for every single gene, but in practice, usually the candidate number of genes we are dealing with can be 1,000, 2,000, 8,000, so that's a huge number. And when we are talking about specific pathway, usually only 20 genes, 30 genes, or 40 genes, so at that kind of scale. So you can see that we are really just trying to fish a very small number of signals from a large pool. And if we are doing this for every single gene, we are kind of wasting, the energy in some way. So what we are doing here is this. So this is a brief description about our method. So we have a regional gene expression matrix. So we have genes in columns, and then we have these experiments. And we are doing a random sampling of the genes first, get a subsample, and then we random partition the matrix into matrix X and Y. And then we apply sparse canonical correlation analysis to these two matrices. So the purpose of doing that basically is to get a linear combination of genes in X and the linear combinations of genes in Y. And those two linear combined genes will show a very strong linear relationship. So you can see that through this analysis, if we are successful, we are getting the strongest linear relationship of a very small set of genes out of all of these genes. So this is the brief idea here, and we are repeating this many, many times by doing many, many random partitions, and also doing many, many random subsamplings. And eventually, we will aggregate all of the linear relationships we have identified together. And the values in the list C here, so C here will be the aggregated coefficients of the linear regressions, and will tell us so roughly what genes are showing strong linear combinations with each other. Some kind of advantages of this idea. So first, what's mentioned here first is really regarding the actual algorithm we are using to perform this SCCA analysis. So one advantage of this is actually we are able to take into account some prior information. So like if there is a target pathway to work on, and we do know a small number of genes belonging to that pathway, then we can take into account that information in the penalty term, in the SCCA, estimation step, and to guarantee that those genes will be included, in the final results. So since SCCA is really a critical tool in this application here, so I just mentioned briefly about what version of sparse canonical correlation analysis we are using. So it's basically based on the paper by written additive threading in 2009, but we didn't use the algorithm from that paper. We used a modified algorithm. So in that paper, they claim that they have a better running time and performance. Okay, so here is a summary flow chart, for the method. So I just want to remind you again what we are really doing. So we have the original gene expression matrix, and we sample columns. So that is basically the subsampling of genes, and we get this subsample of gene expression matrix, and then we do the random splitting of the genes into two sets, and then we will get two submatrices. These are disjoint. And then we run SCCA, sparse canonical correlation analysis, and then we will get unsigned weights. Those are the coefficients for the genes in the linear regression, output from this SCCA program. And here then we are repeating this many, many times. So here is a loop here, and after those kind of many times, then we can get a aggregated measure of the coefficients associated with each gene. And we are also repeating this many, many times, by doing many, many subsamplings. Okay, so eventually we will get this A bar, the adjacency matrix, or the edge similarity matrix, that we can make use of to do the community detection. Okay, so this is the method here. Since we are doing a lot of random partitioning and subsampling, then you may ask, so why those kind of procedures are useful? So actually, subsampling is a critical step to help us identify multiple pathways if there are. So here are just some simple simulation study. We have two pathways involved. One is stronger, the other one is weaker. If we do not use subsampling, we can always identify the stronger, but the weak one will never be identified. That's reasonable because remember that we are running the sparse canonical correlation analysis, and each run of that is looking for the strongest linear combination of genes, in those two subsets of matrices. So therefore, if we are not doing the subsampling by weakening some kind of signals in a strong group, we would never get the pathways with a weaker signal. So subsampling helps us to identify weaker functional groups. And also, subsampling helps the identification of overlapping functional groups. So this is the A-bar matrix we have, so the similarity matrix. So you can see that from here, we can get this overlapping structure very clearly. Okay. So some remarks on this work. So we think this work is conceptually interesting, since it's making use of the aggregation measure of gene partial correlations when the correct conditional set is unknown. So you can see that for the aggregation here, and for the every partial correlation we got here, it's just a partial correlation, under a part, or under, partial order. In other words, we are conditioning on, in most situations, just a subset of the correct set of genes. Okay, so basically, eventually, for the aggregate measure here, is not really a true estimation of the underlying partial correlation. It's just, helpful, to reflect what is the relative level of the, true partial correlation. And asymptotically, it really helps us to separate the genes belonging in the same pathway and the genes are not. And we can prove that asymptotic consistency, for our algorithm under certain conditions. And also the method is very flexible to incorporate the prior knowledge, when they are available. Of course, further investigation is needed on the SCCA procedure and also the community detection methods for the follow-up studies of the S-weight matrix. Okay, so next one I want to talk briefly is related to the local measures. So here is that we are talking about a collection of many experiments. But the relationship, and one example can be we have a very long time series. But the true interaction or the true activity is only happening in a sub-region of the time domain. And what we can do in that case. So since we are talking about really local behaviors of genes, so we want to consider local interaction measure. So the idea here is quite straightforward. So if the set of data we are getting is very heterogeneous, then we can try to define a general co-expression measure by aggregating the interactions across all subsamples of a smaller size. So which means that if the global measure is not the thing we are looking for, if we are caring about the local interactions, we can just try to get all of the local interaction measures, as many as possible, and then aggregate them together, to see what the behavior is. So generally, the idea tells us that we can consider the measure of, as this W. So F is a local measure, and this is one gene X, this is another gene Y. So we get a subsample among all of the samples, for gene X, and the corresponding subsample for gene Y. And then we can measure, locally their relationship. And we are putting all of those local measures together. And if it's true that in some local domain, the interaction is happening, then this W will show a departure from the non-distribution. The non-distribution is under the assumption that there is no local interactions at all. And as an initial effort, we consider the simple F. So like the initial F functions we considered as just the indicator functions, comparing the rank patterns of subsequences. And here are more details here. So the first statistic we considered is called this W1 here, is matching contiguous subsequences of length K. So K here is length of the subsample or subsequence, and N is the length of the original sample. This is especially useful when we consider time course data. So we want to respect, the order of the samples. And then we can define the local measure just by checking whether their ranking of expression is consistent or not in a sub-contiguous sequence of length K. And just as a very simple example, let's look at this. So we have two genes here, X and Y. So originally the sample size is 5, and we first transform the expression values into ranks. And the subsequent sample size is 3. So in this case, what is W1? So we check the first contiguous subsequence. So this is a match. Why? Because at the third position, so this shows in the largest rank, out of these 3, and the first position showing the smallest rank out of the 3. So this is a matched pattern. And this is another matched pattern, right? So this is the largest out of 3, second, third. So this is another matched pattern. And in this case, and the last one, if you look at the last 3 here, that's not a matched pattern. Okay, because this is the largest, this is the largest, so that's not matched. So in this case, W1 is 2. So you see that this is a really simple statistic, counting some local or consistent behavior. And similarly, we can define W2. So W2 here is for the case when we do not have to respect the order of the samples. So in many situations, we are not dealing with time course data. We may be just dealing with data coming from many different conditions. And then we don't really have an order, to respect. In that case, we can just randomly select subsamples from the original sample and then calculate the match of the local pattern. And that's what we have been doing here for W2. So the running time of this, of course, it will be longer than the W1 since we are counting more. And by using dynamic programming and this Bayesian idea here, actually the running time is not that bad. So it's n log n. So k here is the sample size. That can be ignored. So the running time is not that bad. So in the previous case, actually the running time is about n. Okay. Okay. So I wouldn't tell about the details about how to count this, but basically we have this W1 and the W2 as the initial statistics we can explore, for the performance of assessing some local interaction activities. And we do have some theoretical results related to these statistics. So we have their asymptotic distribution when the subsequence is not too long. So if the subsequence is too long, we still have a theoretical gap there. And we can also compute the power of the normalized statistics on the different scenario. And we considered especially 2 very interesting scenarios, for us. One is the case that we have a time series. So we have 2 genes. 2 genes have expression measurements, across time, but only in a sub-region of the time. So like M here, less than N. So the 2 genes are dependent, tightly. So we have that. We say that is partially coupled, time series. And we also consider the case when the 2 genes are following some kind of perfect function, but that function is not monotonic. It is actually following a D strictly, monotonic pieces. So basically, you can see that when D goes to infinity, we are roughly considering, almost every smooth function. Okay, and we can show that so the power is asymptotically optimal, under these scenarios. We also have some simulation studies to assess the performance of the statistic. So this is like in the linear case. So we do have some noise of the data. So X is for gene X. So Y-axis is for gene Y. So this is just a scatter plot of the expression values of 2 genes. You can roughly see a linear pattern here. And the performance was displayed in the left figure. So the x-axis is showing the increase of the outliers in the data. The y-axis is showing the power. So you can see that for this very simple pattern here, most of the methods are working pretty well. So only actually our T1 is not working because T1 is respecting the order of the samples. But in this case here, we don't really have to respect the order of the sample. So that's why it's having some disadvantage. And not surprisingly, so Spearman-Rank correlation is working quite robustly, in this case. And because of the noise, so Pearson correlation is not working as well. Yes. This is the situation when you see that for the 2 genes, so their expression profiles are having this quadratic relationship. And again, we have a lot of noise. Actually, if I don't tell you there is a quadratic relationship, you may not even notice it, right, because of the noise. So there is a quadratic relationship here. And not surprisingly, now, so Spearman-Rank correlation and the Pearson correlation, are not performing well because they were not designed to capture this kind of relationship. But for a lot of other methods, So MIC is this maximal information coefficient method. So that was a method coming from the science paper a few years ago. MI is mutual information. And this is Renyi correlation. This is distance covariance. This is Hofstee distance. So you can see that they are pretty good at capturing some general dependency between variables. And this is another situation that when the relationship is not functional. So the two genes here, you can see that in some cases, they have this negative correlation, and in some cases, they have this positive correlation. But overall, when you are putting the sample together, that's not a functional relationship. And in this case, a lot of methods, become not really performing well again. So you can see that, in this case, Spearman and the Pearson, of course, they were not designed for this case. They cannot work well. But interesting thing is that the Renyi correlation has been working, and also mutual information have been working. Both of methods have been working quite consistently well in the situations we have studied. And our T2, so which is not respecting the order, is also working, quite well. Okay, so this is the last case is actually for the case designed for our method. So we have partially coupled time series, and in this time domain here and in this time domain here, we have very strong relationships between the genes. So here we have very strong negative correlation. Here we have very strong positive correlation, but no relationships in other time domain. And you can see that because the relationship is only happening in a very small side of time points. So if you just look at the scatter plot of the two genes, you wouldn't be able to see anything. So no relationship has been showing. And that basically means that for all the other methods we have been considering, they would fail in this case. That's reasonable because they were not designed, for this situation either. And our method here for both T1 and T2, they stand out. So they are not as powerful as in the previous situations, just because this situation is more challenging, but they are definitely much better than all the other methods. And we applied it to some real data, and for the real data, this is just one case for two genes. They are known to be biologically, related. And for the data here is involving 4 time courses, and only in 1 time course, actually the two genes are showing some co-expression pattern. And using other traditional methods like Pearson correlation, Spearman correlation, so that two genes, will be missed. But our method is able to identify that. So we basically think our method is really complementary to the existing methods to detect, gene relationships in this kind of special case. Okay, so last one, just 1 minute. I will talk very briefly about this ongoing work. As I said that I won't have time to talk about this in details, but I do want to mention a new concept we are trying to make from this study. So you know that there have been a lot of active research going on for EQTL studies. So that's the genomic loci contributed to the changes of gene expression. So now we want to define something in an analogous way, but it's not for the change of gene expression, it's change for the gene co-expression. So we want to study basically the change of the networks, relative to the change of genotype. And in some preliminary studies, we do see some very interesting results coming from that already. So like, in these two different genotypes, we do have a lot of individuals belonging to these genotypes. So the correlation of the genes have been different. So this is ongoing work. And I hope in the future I will have opportunities to report more on this work to you guys. So finally, I want to acknowledge all of the co-authors on this work, on the work I have mentioned. So like Prof. Haiyan Huang and Peter, so they are in the audience. And there are also a lot of other, either my PhD students or postdocs, or my biologist collaborators, are involved in this work. So finally, thank you everyone for your attention. Yes?
In this first part of your work, why was it important to express into groups?
Because we are looking for a very strong linear combination, of genes, right? So, but after the random partition, we are able to apply the sparse canonical correlation analysis. So the sparse canonical correlation analysis is designed for that. If we have two sets, you can find a linear combination of one set, a linear combination of the other, and then.
So standard CCA is known to have the problem that when the dimension is high, it overfits, giving very spurious directions. What I'm wondering is, are you avoiding that because of your subsampling or because of the sparsity?
Because of sparsity.
Okay, so you think you can handle pretty big subsamples then?
Yeah. Yes.
So for classical CCA, and you have to normalize your data first.
Yeah.
And
this one we have to do the
now you're
normalization.
not doing it, right? And so you're supposed to, you have your x, you have your y, and you're supposed to use in the same sigma x to power minus 1/2 to normalize it. Now you're not doing it, and is that going to have problem?
That's a good question. So we didn't really take into that in details, but for that project, we have been working on microarray data. And for the microarray data, after the traditional normalization method, usually for each dump of the data, it's following normal pretty nicely. Yeah. So, yes?
And the splitting, once you average those A's and B's over many samples, do you have some intuitive interpretation of what that average means?
So you can see that for each round, we are getting a linear combination of genes. And because of the subsampling and also the random prediction we are getting, we are expecting it can be just a subset of genes of the two sets we are looking for. And so basically for that, you can see roughly the high numbers will tell us the correlation is strong, and low numbers, the correlation is low. And if we are aggregating, many rounds of such things together, for the genes, they are consistently belonging to some identified linear relationships, and their coefficients will be added up. And that, coefficient will stand out eventually.
Do you interpret it as how many of those genes are strongly correlated with the other one, like small subset, a big subset? Is that
so in practice, usually we are expecting like only a very small number of genes are related, like 10, 20, or 30 out of like 500 or 1,000, something like that. Yeah. Okay. More questions?
That's fine. Speak very good.